Viral vectors have evolved over millions of years to possess intricate biological machinery, allowing them to penetrate host cells and navigate directly to the nucleus. On the left side of this animated diagram is a typical viral vector covered in specialized receptor-binding proteins. On the right is polyethyleneamine, or PEI, the widely recognized gold standard synthetic alternative for non-viral gene delivery. PEI possesses no evolved proteins for active intracellular transport or environmental sensing, entirely devoid of capsids and targeting motifs. Structurally, it exists simply as branched polymer chains, its functional capacity derived entirely from repeating primary, secondary, and tertiary amine groups. Lacking specialized targeting ligands, PEI relies on the interaction between chemical thermodynamics and host cell cycle dynamics to advance the plasmid toward the nucleus. This journey occurs in two distinct phases. First, a brute force physical chemistry breach into the cell interior, followed by a period of opportunistic biological integration. Effective non-viral transfection is a product of these physical and chemical exploitations rather than the mimicry of viral biology. The initial formulation challenge is taking long, extended chains of negatively charged plasmid DNA and packing them tightly enough for microscopic delivery. At physiological pH, the amine groups positioned along the PEI polymer backbone become heavily protonated, generating a highly dense positive charge. This dense positive charge binds electrostatically to the negatively charged phosphate groups of the DNA backbone. The interaction neutralizes the DNA's negative charge, forcing the loose nucleic acid chain to violently collapse and condense in upon itself. This newly condensed, tightly packed spherical miniparticle is defined as a polyplex. During formulation, an excess of PEI is intentionally used to ensure that the final polyplex maintains a net positive surface charge. This net positive charge acts as a physical repellent to other particles, preventing the polyplexes from aggregating into useless clumps while in solution. We see the secondary function of this charge when we shift perspective to the exterior of a target host cell. The plasma membrane is heavily anchored with negatively charged proteoglycans and sialic acid residues. When the positively charged polyplex makes contact with the cell membrane, it creates an immediate, nonspecific electrostatic attraction. This simple electrostatic binding stimulates the host membrane to invaginate, engulfing the polyplex into a membrane-bound vesicle called an early endosome. The internalization process is driven by this nonspecific electrostatic attraction, leading the cell to engulf the polyplex as a byproduct of membrane interaction.
The polyplex is now trapped inside an early endosome, facing an immediate biological threat. The host cell intends to mature this vesicle into a lysosome to digest the foreign material. The host cell actively acidifies the vesicle. VATPase pumps push protons inside to rapidly lower internal pH. But PEI executes the proton sponge effect. Unprotonated amines on the chain absorb incoming protons, causing the pH meter to freeze. Because PEI contains a vast number of these secondary and tertiary amines, it acts as a massive chemical buffer against the acidification. This creates a strict thermodynamic trap. Protons enter, but internal pH remains stable, disrupting electrical balance. To maintain neutrality, the host cell blindly pumps massive amounts of chloride ions into the endosome. The thermodynamic consequence of this sudden ion accumulation is a severe osmotic gradient.
The physical reaction is immediate. Water is rapidly driven into the vesicle's lumen to balance the ion concentration. The continuous water influx causes severe internal swelling, stretching the lipid bilayer to its absolute mechanical limits. The internal osmotic pressure inevitably exceeds the physical tolerance of the membrane itself. Ultimately, this triggers the explosive lysis of the endosome, cleanly ejecting the polyplex payload directly into the cytosol. This escape mechanism leaves the polyplex intact, but it is now entirely exposed in a new cellular compartment. PEI escapes enzymatic destruction by turning the host's own digestion mechanism into an explosive thermodynamic bomb. Now in the cytosol, the naked polyplex faces a highly hostile environment, immediately exposed to the cell's autophagic degradation networks. Without rapid intervention, these defense systems will dismantle and destroy the foreign plasmid within seconds. The payload survives due to the immediate, unintended intervention of a host protein known as barrier to autointegration factor, or BAF. BAF binds tightly to the foreign plasmid DNA almost instantly upon its exposure to the cytosol. Once bound, BAF executes a secondary action, rapidly recruiting host endoplasmic reticulum membranes directly to the site of the plasmid. Looking at the schematic cross-section, you can see the result. The plasmid is encapsulated within an ER-derived double-lipid bilayer, forming a distinct nuclear envelope-like enclosure that temporarily shields the DNA from degradation. A totally synthetic vector ensures its structural stability in the cytoplasm by hijacking the host's own structural defense proteins. The ultimate physical barrier to gene delivery is the nuclear envelope, which remains fully intact and impenetrable throughout interface. Because PEI polyplexes completely lack nuclear localization signals, the tightly regulated nuclear pore complex is completely inaccessible to them. Structural entry into the nucleus is physically impossible until the host cell naturally progresses into mitosis to undergo cell division. Entry begins during prometaphase, when the host cell naturally breaks down its own nuclear envelope to divide its chromosomes. As the true nuclear envelope breaks down, the BAF-induced ER protective shell around the cytosolic plasmids also disassembles. Visually, the large, immobile cytoplasmic plasmid clusters disperse into small individual puncta dynamically distributed near the mitotic spindle apparatus. The critical, fleeting window for actual nuclear integration occurs as the daughter cells begin to form during telophase. This diagram outlines two capture pathways. Left, mechanism A, direct binding. Individual plasmid puncta bind directly to bare chromatin masses before the envelope seals. Right, mechanism B, membrane fusion. Encapsulated plasmids fuse directly with reforming nuclear envelope precursor membranes as they seal. The physical journey is completed when the host cell finishes division, safely enclosing the plasmids inside the fully sealed G1 phase daughter nuclei. Once inside, the host cell's transcription machinery gains access to the plasmid, driving transcription into mRNA, which is exported back to the cytoplasm for ribosomal translation. The appearance of a functional transgene product at a median timeline of 150 minutes post-mitosis proves that a completely synthetic material can master cellular biology through a calculated mix of thermodynamic violence and patient opportunism.